Hello students, welcome back to a new video of Sunanda's tutorial. So this video is especially for CHSE students, plus 2 second year students, those who are going to appear their board examination. So for them, for those students, from now I will upload each and every chapter of botany. So the first chapter is heredity and variation. So here I will discuss the whole chapter in one video and important points of each chapter. So basically it is a revision video. So let's start. At first, some terminologies related to genetics. At first, what is genetics? Study about genes. What is gene? Functional part of a DNA. Okay. Next, what is dominant? The character which expressed in F1 generation. What is F1 generation? Students, what is F1 generation? F1 means when parent 1 is crossed with parent 2, the first hybrids, those who appeared in first generation, are called as first filial generation. Okay, and the character which is expressed in this first filial generation is called as dominant character. Okay, next, what is recessive? The character which is masked by the dominant gene is said to be recessive. Okay, next, what is allele? Allele means a pair of genes controlling the same character. Suppose the tallness of a plant is controlled by capital T, capital T. So this is one allele, this is another allele. Or T, small t. This is one allele, this is another allele. Okay. Basically, two alternative forms of same gene. Two genes basically control the height of the plant, but they are two forms, the two alternative forms. One is tallness, one is for dwarfness. Capital T is for tallness, small t is for dwarfness. Then what is homozygous? Homozygous means it has two same copies of same allele coding for a particular trait. Suppose for tallness, homozygous condition is capital T, capital T. Okay. Homozygote produce only one kind of gamete because they have only one type of gamete, only capital T. So they produce only one kind of gamete. Okay. They produce only one kind of gamete. Okay. Next, both genes of homozygote are dominant. The genotype is called as homozygous dominant. When both genes or both allele of a gene are dominant, are called as homozygous dominant. For example, tall parent, capital T, capital T. When both genes of homozygote are recessive, the genotype is called as homozygous recessive, small t, small t. Dwarf parent, small t, small t. Next, what is heterozygote? When in individual carrying non-identical gene. For example, capital T, small t. The phenotype is tall, but genotype is in heterozygous condition, that is capital T, small t. Here the dominant character is expressed, but the genotype is capital T, small t. Okay, next. The hybrid resulting from cross between two parents differing in one character is called monohybrid cross. In a cross, when we observe only one character, it is called as monohybrid cross. The F1 or first filial generation plants of Mendel's monohybrid experiment is called as monohybrid. The hybrid resulting from a cross between two parents in two character called dihybrid cross. When we observe the heredity or the transmission of only one character from their parents to their offspring, it is called monohybrid cross. When in a cross we observe two character of the parents involving parents It is called dihybrid cross. Okay. The F1 plant of Mendel's dihybrid experiment is a dihybrid. Okay. So phenotype and genotype. What is phenotype and what is genotype? What we observe or the external appearance of any individual is called as its phenotype. Then what is genotype? Their genetic constituent is called as genotype. Okay. So in monohybrid experiment, you have already studied about monohybrid experiment. Monohybrid experiment means when Mendel crossed a pure tall plant with pure dwarf plant, the first filial generation, only in first filial generation only tall plants appeared. Okay, so in this way it is called as F1 generation. then by selfing they he got plants with 3:1 ratio. That means 3 tall plants and 1 dwarf plant. So 3 tall plants and 1 dwarf plant. Okay. So these are genotype. Genotype means capital T, capital T, capital T, small t, capital T, small t, small t, small t. These are genotypes. And what is the phenotype? Phenotype means these are tall, this is dwarf. So these are phenotype. Okay. Next, in Mendelian experiment, the ratio 3:1 means 3 tall and 1 dwarf. This ratio is phenotypic ratio. The genetic makeup of organism called genotype. The genotype of pure breeding tall plant is capital T and dwarf plant is small t, and the genotype of hybrid is capital T small t. Okay. Next, the transmission of character from the parent to offspring is known as heredity. Some of the characters of an offspring appear totally different and do not seem to belong to either of them. This difference is called variation. So in transmission, so generally a character transfer from their parents to offspring. So that is called as heredity. But some offspring difference appear totally different, called as variation. They vary in characters. Okay. So what is the reason behind it? Due to random fusion of gamete, male gamete and female gamete, due to crossing over during meiosis, gamete formation. So these are the cause of variation in offspring. Certain organisms inherit a set of character from single parent without involving gametic fusion, without sexual reproduction. So asexual reproduction in case of asexual reproduction, they are called as monoparental. So in asexual reproduction, gametic fusion is totally absent, and the offspring which are produced from a single parent called as monoparental. The progeny would inherit the same set of characteristic of parent, would not be different from it because it is produced from a single parent. So there is no gametic fusion. So no variation or recombination. Okay. Such a genetically uniform population originating from a single organism is called as clone. Okay. Next. On the other hand, higher organisms reproduce sexually, producing male gamete and female gamete. They are called as biparental. Okay. Next, the study of this heredity and variation is called genetics. This term was proposed by William Bateson in 1905. However, the first and the most important contribution to genetics appeared as early as 1866 when Gregor Johann Mendel published his work in latter half of 19th century. He is known as the father of genetics. Remember his name, Gregor Johann Mendel. Okay. Then Mendel's life history. So he was born in 1822 in Hessendorf, which was a part of Czechoslovakia. His father had great interest in plants, which influenced Mendel, and in 1847 he came to Brunn to study for priesthood at Augustinian monastery. Later he went to study science at University of Vienna. Mendel taught at Brunn Modern School for about 14 years. Okay, he began his genetic experiment there using pea plant in 1856 in the garden at that monastery. These experiments took 8 years for completion. So imagine Mendel did his experiment for 8 years. Okay, so the result of his work were presented in a research paper entitled Experiments in Plant Hybridization and read before Brunn National History Society in 1865. So this is about Mendel's life history, who is the father of genetics. This paper was subsequently published in the Proceedings of the Society in 1866. It contained Mendel's hypothesis concerning the mechanism of inheritance. The paper remained practically unnoticed during Mendel's lifetime. He repeated this experiment on another plant named Herasium and some other plant material, however, failed to repeat his observation. He later became abbot of Brunn Monastery in 1871 and died in 1884. So this is the whole life history of Gregor Johann Mendel. Then rediscovery. So during his lifetime, his experiments was unnoticed. So now after his death, his work was rediscovered by Karl Correns, then Cermak and Hugo Dribris. Okay, so they published Mendel's work again in 1900. Okay, so this is totally about history. Now, what are the major reasons for Mendel's success? How he got the success during his experiment? At first, he selected pea plant for his experiment because there were very several variety of pea which show distinct alternative traits. That means the different varieties show different characters, different visible characters. Then the plants were easy to cultivate. We can easily cultivate pea plant. The short lifespan was also useful in getting quick result. So we can get quick result when we did our experiment in using pea plant. The flowers are self pollinated, so we can manipulate for we cannot manipulate the cross pollination, because in cross pollination agents are involved, cross pollinating agents, but in monohybrid cross we can manipulate in cell pollination. Next, the reproductive structures are completely enclosed by petals. This helped Mendel to maintain varieties with same character generation after generation. The varieties could be artificially cross pollinated. The hybrid thus produced were fertile. Okay. Next, working method. Mendel got the success was due to his meticulous planning. He properly planned his experiments. Okay, he carried out preliminary investigation, carefully planned in a way only one character was studied at a time during monohybrid cross. Special care was taken to avoid cross pollination. He kept accurate results, accurate records of all his experiment. He also obtained sufficient data so that it could be statistically analyzed, because at first he was a mathematician, so he observed every data and analyzed every data statistically. Okay, Next, the hybridization technique. At first, emasculation and bagging were the techniques used by Mendel. Emasculation means at first he removed all the male part of a flower and made it a female flower. He made one plant, a female plant, by removing its male part. Then another plant, he made another plant as male plant by removing the female parts. So that is called as emasculation. And bagging means after that, after producing male plant and female plant, he completely covered it using a bag of plastic to avoid cross pollination. Okay, so these are the techniques of hybridization. Okay, if the flower is bisexual, removal of anther from flower bud before anther dehiscence is called emasculation. Then from suitable pollen grain were collected from a suitable plant and sprinkled on emasculated flower. Then after dusting, the flower have to be covered with bag of suitable size made up of butter paper to prevent contamination of its stigma with unwanted pollen by cross pollination. It is called as bagging. Next, so these are the characters Mendel selected. Okay, next, then monohybrid experiment. I have already told you about this. One character was chosen for his experiment during monohybrid experiment using pea plant. Mendel selected two pure breeding pea plant, one with tall stem, one with dwarf stem. These plants are the parents and pure breeding varieties. The parent pure breed tall and dwarf plants were crossed. The seeds collected from these plants were sown. The plants which grew from these seeds are first filial generation. F1 generation were all tall plant, and then they were interviewed with each other. Then all the seeds of F2 generation or second filial generation were collected and raised. In F2 generation, two types of plants were found, one tall and one dwarf. So in the ratio 3:1, 3 tall and 1 dwarf, and 1 pure tall, 2 heterozygote tall, 1 pure dwarf. In this way, with genotype 1:2:1. Okay, next, in F2 generation, 4 possible combinations TT, capital T, capital T, capital T, small t, capital T, small t, and small t appear. So the dominant gene is capital T. Okay, so this is the diagrammatic representation. Pure tall, pure dwarf, these are gametes crossed to get F1 generation with capital T, small t, then cell pollinated to get capital T, capital T, capital T, small t, capital T, small t, then small t in 3:1 ratio, and genotypic ratio is 1:2:1. Okay, remember this. So it is all about monohybrid experiment. It is the part 1 video of your chapter Heredity and Variation. In part 2 video, I will discuss the rest part of this chapter. So please stay tuned, and if you liked it, please hit the like button, share with your other friends, and please subscribe to get more videos. I will discuss each and every chapter of biology in this channel. So please stay tuned. Okay, thank you.